We're going to do Chapter 7, Environmental Nutrition, Ecology, and Growth of Bacteria, environmental stuff. All right, so start off with microbial nutrition. Nutrition is just acquiring substances from the environment that the cell uses to make all the products they need to grow and survive. Essential nutrients are ones that the organism cannot create on its own from what it gathers. Right. So, or excuse me, yeah, the ones that they have to have. Two categories of these are your macronutrients, which are large molecules and they need them in large quantities. Your macronutrients are proteins, carbohydrates, nucleic acids, and lipids. The PowerPoints give you the proteins and carbohydrates. Because from these, the metabolism of the organism, the cell, can break them down and build them up and make different stuff out of them. So that's the macronutrients. Micronutrients are ones that the body cannot, or the cell cannot make on its own. So it has to acquire these from outside. Sometimes it's vitamins they have to get. They're giving you as examples of some metals that they can't make. For like humans, we have there's 20 amino acids, and 11 of them, actually 9 of them are what we call essential amino acids. In other words, amino acids that our body cannot make, so we have to consume those amino acids. But it's going to vary for each species. But again, those are ones that we have to consume. We need them in certain amounts. Those are your micronutrients. All right, your nutrients are organic for the most part. Organic means that it contains carbon and hydrogen. All right, and it's usually made from other living things, but not always. There are some organic compounds that occur naturally in the environment that without any life around them. I'm giving you some examples. So, like methane, CH4, see it has the carbon and hydrogen, so it is an organic compound. Oil is an organic compound. It's basically a bunch of carbon and hydrogen, but it's an organic compound because it has those two things. So again, we have others, and they're giving you these. There's those macronutrients: the carbohydrates, lipids, proteins, and nucleic acids. Those are all organic nutrients. Inorganic nutrients lack either carbon or hydrogen, right? But they can be made up of anything else. Good examples are like oxygen. Oxygen's O2, no carbon, no hydrogen. Water, hydrogen and oxygen, but no carbon. So it's an inorganic molecule. And we have to have, all life as we know it has to have water. So there are inorganic nutrients that you have to have also. Of a cell, by far the majority of a cell is, because again, all life as we know needs water, so by far the majority of a cell is water, 70% of it. That's the inorganic, that's an inorganic portion of it. Most of the organic portion of a cell is made up of proteins, but there are some other stuff. Now, if we look at all of it, 96% of a cell is made up of these 6 elements: carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. And mainly it's the carbon, hydrogen, oxygen, and nitrogen. Phosphorus and sulfur in smaller amounts, but those 6 make pretty much everything. And then you have like zinc and copper and nickel and magnesium. Those are all in small amounts of it. So these things we look for life. So our carbon sources, where do we get carbon? All right, a heterotroph must obtain the carbon from an organic form. All right, and again, we come back to those main macro molecules: proteins, carbohydrates, lipids, and nucleic acids. For the most part, those are created by another living organism. We can get like amino acids and some nucleic acids or nucleotides, I should say, from inorganic means, but by far the majority of the, just because they occur naturally, the majority of these are from organic made from other living organisms. An autotroph, in this case, is one that uses an inorganic carbon, so CO2, because there's no hydrogen in there, so that makes it inorganic, and they use that as the carbon source. All right. Now I said proteins over here, but amino acids don't necessarily have carbon in them. So again, they're getting carbon from an organic thing, and again, these are using carbon dioxide as their carbon source, so that they do not depend on other living organisms. All right, so these compounds that cannot be synthesized because they lack the genetic and metabolic mechanisms to make them. These are what we call growth factors, and again, those are essential amino acids I was mentioning earlier. Like give an example, E. coli doesn't. It's a very versatile bacteria. It doesn't need. It has about like 5,000 different components in it. And it can make most of them from just a few things. All right. What it can't make, then those will be growth factors that are going to affect how it grows. And so then that's what it needs. So we talked about our carbon sources. Now we need to look at our nutritional types. Our energy source. We have two types of energy sources. We have our chemotropes, which are the ones that use chemical energy, chemical compounds, again, to get their energy for life to perform all their functions. And we have what we call the phototrophs, those that use light, because photo is light. So they're going to get all their energy through photosynthesis. And here's a breakdown of things. Now we mentioned the energy, which is the chemo or photo. And we've also mentioned the carbon source, which is auto or hetero, and these can be combined together. So like a photoautotroph, their energy is sunlight, and since they use carbon dioxide because they're an auto. So right here, organisms, you think of algae, plants, cyanobacteria for these. Now we have some autotrophs that use chemical energy. But these are only bacteria and archaea, and these are ones, there's not a lot of these things, but these are ones that use chemical energy or chemistry for their energy, and they use inorganic carbon for their carbon source. Now on the other side, for our organic molecule, our heterotrophs, the ones that use organic carbon, we have both types. Most of these are chemoheterotrophs. In other words, they use something other than sunlight for their energy source. Most of these, again, are going to be using some kind of organic compound, but not always. So they're using something to get their energy, and they are using an organic carbon. So they are consuming other carbon made from other organic other life. And they can break these down into some saprobes. These are ones that consume dead material. It's mainly fungi and bacteria, but there are some protozoa that do it. These are our decomposers. That's what a saprobe is. And then we have some symbiotic ones, and these are ones that live on another organism. So these are our parasites, most microbes, or not, I shouldn't say most microbes, some microbes. Now we do have some heterotrophs that use light as their energy source. Again, there are not very many of these. And these are known as purple and green photosynthetic bacteria. But they do use an organic carbon source. They just use light energy instead of chemical energy. For our photoautotrophs, in other words, the ones that use light energy and an inorganic carbon source, we have a couple things: oxygenetic photosynthesis and anoxygenic. Remember, an A before a word means without, so some of them use oxygen to get energy. Some of them you go without using oxygen. They'll use like hydrogen sulfide. Well, that's an energy source, but they'll get their energy some other way, using some other compound, hydrogen sulfide, There's one way to do this with photosynthesis. We have our chemoautotrophs. These usually, again, surviving completely on inorganic carbon, also known as lithotrophs. They give you some examples. Your methanogens. Most of these are all RK, and are they all RK? Most of them are RK. Most of them lived deep under the ocean, underground. They produce methane gas under anaerobic conditions, in other words, without oxygen. There are some that live in the intestines of animals, humans even for that matter. And again, they produce methane. And they're showing you some right here. All right, the energy sources. So again, majority of chemoheterotrophs use aerobic respiration. In other words, they need oxygen. All right, so we have a couple of categories underneath here. Again, saprobes, these are free living ones that decompose or eat dead organisms. Now, they are not strictly that way. They can be what we call an opportunistic pathogen. In other words, under the right conditions, they can start to live on a living organism. In other words, cause an infection on a human or some other organism, instead of just waiting for it to die, then start to grow on it. But then again, that is opportunistic. It's got to be the right conditions for it. Then we can have what we call facultative parasites, which means that they're causing a disease when they're doing this. All right, so let's go to the parasites. Again, deriving nutrients from a host. It causes some harm, not a lot necessarily. Now, if it is definitely causing harm that can kill you, then we call those pathogens. There's a slight difference between a parasite and a pathogen. A parasite will live on another organism and does cause it harm, but not necessarily a disease or doesn't necessarily, it's not trying to kill it. Pathogens, I don't want to say are trying to kill it, but they cause diseases and they can cause death. Parasites don't normally do that, I should, but again, it's very ambiguous. Then some are obligate parasites. In other words, they have to live on or in an organism to perform all their functions. There's not a lot of these, but there are some out there. Viruses are all obligate parasites, but they're technically not alive, so I'm trying not to include those in this example. Rickettsia is an obligate parasite. That's a bacteria. Has to be in a host to perform all the functions. Some other environmental things here. We need movement across membranes. This is diffusion, osmosis kind of stuff. Your passive transport, and then active transport, which is energy. Diffusion is moving any molecule from a higher concentration to a lower concentration, no energy. Osmosis is diffusion, but specifically talking about water. Facilitated diffusion is diffusion that uses a carrier. It doesn't require any energy still because it is diffusion, but it's a carrier that helps it along, helps it to process along. Then active transport uses energy, and there's a couple different kinds of this. You have your group translocation, which is it's using energy, but it modifies the substance so that the substance can be used right away. So it saves a little bit of energy that way. And then you have a couple of things to use bigger objects in their endocytosis and pinocytosis. Exocytosis, getting rid of it. So diffusion, again, you put it in your sugar cube there, so it's really concentrated, and you see it right here, and it slowly moves out because there's no sugar right here, so it's going to end up moving out that way again. Simple diffusion. Osmosis, so it goes by the concentration of the water molecules or of the solute that's dissolved. The red ones here cannot fit through the membrane, so it's affecting the concentration of the water in it, and outside there's no red molecule, so that's like 100% water. So if you look at the top of the water here, you'll see that the water is generally moving in because the water level gets higher in it, and it's doing that to dilute the solute inside of it. So that's osmosis again, because there's a higher concentration outside than inside, so you have a net movement going into the substance. These solutions, we call them isotonic, hypotonic, or hypertonic. Isotonic, which means the inside and the outside are the same. Hypotonic, the outside concentration is less than the inside concentration, so you have a net movement of water going into the cell. And on a hyper concentration, the concentration outside of the cell is higher, so you have a net movement of water outside of the cell. Now this is one that has a cell wall, so it's not going to, doesn't change much. So your isotonic, again, the movement of water is equal going in and out of the cell. On your hypertonic solution, hypotonic solution, more water is going into the cell, and if it doesn't have some kind of means to regulate this, it can lyse. On the hypertonic solution, since more water is leaving the cell, if it can't regulate that, then it will shrink. We call it crenation. Facilitated diffusion, again, there's a kind of a protein in the plasma membrane that helps the molecule move across. Now it moves both directions, in and out. So if you have more on the outside, you're going to have a greater movement in than you will movement out. Again, it doesn't take any energy. Water actually goes through proteins in this way. So technically it's a facilitated diffusion, but it's through aquaporins, or what we call them. But it's a specific type of diffusion. All right, active transport right here. Key thing is using energy. This will go against concentration gradients, or it can be used to move ions, because ions don't like to move across the membrane without energy. So that's where this comes into play. Your group transport it's specific. You have proteins and they're specific to a certain substance. It is using energy, okay? And as this substance moves in, it gets modified to a usable substance that something that the cell can use right away. So it saves a little bit of energy because it's modifying as it comes in instead of just moving it in, then modifying it. The other item is endocytosis. So this is bringing something into the cell. Okay. Two processes. Phagocytosis is using solids, bringing in some kind of molecule. It could be a cell for that matter. Like our white blood cells would do this, even though we're talking about bacteria in this case. They're still bringing some kind of a solid in. And then you can see with these microvilli here, this is used to bring water in. These villi will connect at the top, and now that water, that fluid, that liquid there, is ingested as a vesicle. Then they'll break down the membrane around the vesicle, and that fluid is inside of the organism. Just like over here, you have they'll slowly wrap around it and connect, and then the membrane around the vesicle is dissolved, and that substance is inside of the cell. So that's phagocytosis and pinocytosis, which are specific types of endocytosis of bringing it in. If you reverse this process, we call it exocytosis because it's exiting the cell. So it'd make the vesicle and slowly move it out, and then they would break apart and release it. Same process. We call it exocytosis. This is a pretty good chart, kind of familiarize yourself with things. Up here is all your passive transport, and down here is your active transport. All right, environmental factors that influence microbes. Every organism has a niche, all right? And the niche is the total of all of the adaptations that the organism has to live in that specific habitat, okay? As like, you're not going to take a shark and put it on land because it doesn't have the adaptations to do that. They're perfectly adapted for what they do in their environment, in their niche. Just like we can't go do what the shark does because that's not our environment, it's not our niche. Same concept for microbes. All right, some things that affect these adaptations would be like temperature, oxygen requirements, pH, osmotic, barometric, and we're going to break down some of these. So for temperatures, there's three main categories here for each organism. They have a minimum temperature. So this is the lowest temperature that they can still perform all of their metabolic functions and survive. They have a maximum temperature, which again, just like it says, the highest temperature that they can perform all their metabolic functions and survive. And then we have what we call the optimum temperature. So the ideal one, the one that just like Goldilocks and the Three Bears, not too hot, not too cold, just right. That's what the optimum temperature is. The optimum temperature is close to the maximum temperature because with chemical reactions, the easiest way to speed them up is to add heat. So you want, since metabolism is all about chemical reactions, we need to have them, we want them to go fast enough for life. So the optimum temperature is going to be close to the maximum temperature. Okay? So here, if you look at the chart down here, they show some, like these here cyclophils in blue. This is the maximum, this is the minimum, and notice that the optimum is pretty close to the maximum. Same with all of them. Their optimum is real close to the maximum temperature. All right. So our three basic categories on them are cyclophiles. These are ones whose optimum is below 15 degrees Celsius. So you're talking roughly 45 to 50 degrees Fahrenheit. All right. They can grow at zero, so at the freezing point. But that's, again, at the lower end of them. But they can get below freezing. You notice down here, negative 15 degrees Celsius. You're getting cold. Mesophiles, the middle. All right. These are ones that are going to be near that 40 degrees. So right over here, see their optimum is right here close to 40. What's the human average human temperature? 37 degrees Celsius. So you're up in that range. That's why these are, this is where most human pathogens falls in that area. Then you have your thermophiles. Thermo means temperature. Philes, loves. So we're talking about high temperatures. So these are above 45 degrees Celsius, which is 45 degrees Celsius. You're talking about 120 degrees, and that's their low end. Excuse me. Yeah, that's their low end. And then you've also got your extreme thermophiles. And notice their optimum temperature is like 120, 125. Boiling point of water is 100. So these live in like hot springs, deep thermal vents where the water's really hot, but it's not boiling. It can't boil because of pressure. So yeah, these are really extreme, hence extreme thermophiles. They like really, really hot temperatures. So yeah, remember your cyclophiles are cold, mesophiles are your middle, thermophiles are your high, and then you have your extreme thermophiles. I don't worry too much about the cyclotrophs. Well, I probably should, but I don't put too much of that on the tests. Gas requirements. We're egocentric, so we're everything based off us. That and most things anymore use oxygen. So these are going to be based on oxygen. Now, oxygen can be transformed into toxic properties, and actually, like singlet oxygen, our white blood cells will produce singlet oxygen to kill bacteria. And you find these things in the environment, and organisms that cannot process these have to live away from them. So they're the ones that live in the anaerobic environments. Most cells, though, have developed enzymes that do this. It's a two-step process. Taking either the singlet or the superoxide ion, the superoxide dismutase, basically it's the superoxide. will turn the superoxide ion into the hydrogen peroxide. And so that's this particular enzyme. And then catalase will take the hydrogen peroxide and turn it into water and oxygen. But so they have to develop these methods, these enzymes, to neutralize these reactive chemicals, reactive oxygens that can be dangerous, that are toxic. Again, if it cannot do with this, they're forced to live in oxygen-free habitats. Since oxygen has been in the atmosphere for quite some time now, most organisms have adapted to using oxygen, but not all. All right, so let's categorize them. If they use oxygen so they can detoxify it, we call them aerobes. Now, under the aerobes, we have 3 subcategories. The first is the obligate aerobe. In other words, these have to have oxygen. All right, they cannot grow without it because they're obligated to have it. Then we have some that we call facultative anaerobe, and now these are ones that use oxygen, but if there's no oxygen, they'll still be all right. They can grow without any oxygen. Another one is called our microaerophilic, and these here are ones that require just a little bit of oxygen. So places where we're going to find a little bit of oxygen would be like high up in the mountains or high up in the atmosphere. So that's where you'll find the microaerophilic ones. Anaerobes, these are organisms that do not use oxygen, and we have 2 types of these. We have our obligate anaerobes that cannot detoxify oxygen, so if you put oxygen around them, it'll kill them. So these do not want any oxygen at all. And then you have your aerotolerant anaerobes that they don't need the oxygen to survive, and they don't want the oxygen, but if it's there, they'll still be able to grow anyway. So they're tolerant to the oxygen. Looking at these tubes here, just to kind of review it a little bit. If you notice in this tube, there's nothing growing down here, but you can see some right here at the top. So based on our oxygen requirements, come back over here. Since it's growing at the top here where there's air, we know it can grow with oxygen, and it's not growing down here in this solid where there's no oxygen. So this one has to be an obligate anaerobe. It has to have the oxygen. That's why it's only growing here at the top. And you look over here at this next one. Again, the bacteria is growing at the top, and it's growing down here where there's no oxygen. Right. So this one can grows with oxygen, and if you get farther away, it's not even growing down here where there's no oxygen. So this one here would be a facultative anaerobe. In other words, it wants the oxygen, but it can grow without it. This one here is kind of a trick question, but if you look at it, again, you have bacteria growing up here, and there's quite a bit through here. And as you get lower and lower, it starts getting a little bit thinner, but it can still grow quite a ways. This one also is a facultative anaerobe because it prefers the oxygen. As you can see, there's more of it up here, and it gets further and further away. There's less of it, but it can still grow without it. This one can grow better than this one can without oxygen. That's why it's growing out further. You look at the 4th one, it's really dense down here. You can't even see through it. And then it's real clear right here. Excuse me, and there's nothing at the top where the air is. So this bacteria does not like the oxygen, can't grow near it. So this one here would be an obligate anaerobe. Over here, they're looking at ones that cannot grow with oxygen. That's why they're in the incubator there with the tubes. There's another type in here that some like to have more carbon dioxide. So we call these capnophiles. So anytime you see capno, C-A-P-N, it's talking about something that likes our carbon dioxide. So these have a higher carbon dioxide than what you normally find in the atmosphere. And in a human, the only place like that would be inside the lungs, is where there's more oxygen, or excuse me, more carbon dioxide in our lungs than there is in the atmosphere. There's really not much in the atmosphere. So if we were infected by one of these, it would be in the lungs. We have pH levels. The middle of the pH scale is 7 because it's 0 to 14, so 7 is the middle. And most organisms live right around that middle portion, neutral, so 6 to 8. Those that like high acid, so phials means loves, acid, so they love acid, are going to grow in an acid, and they're going to be at a low pH because low pH is an acid. The alkalinophiles, the ones that love alkaline, are going to grow in an alkaline pH, which means they're going to grow in a high pH. So above 7 is alkaline, below 7 is acid, but usually we're talking like it, lower than the 6 or 8 even, because technically 8 is an alkaline and 6 is an acid. But we want to get further away from the middle. Osmotic pressures. So this is really going to be dealing with salt, because salt's the biggest thing that affects our the osmosis, the movement of water in and out. So halophiles, again, phial means love. Halo is the halides, these are the salts. These love the salt, and they'll live in extremely high salt concentrations like the Great Salt Lake, Dead Sea, places, salt flats. These organisms can live there. Then you have what you call the osmotolerant. Sometimes they're called the facultative halides, because they do not, they prefer the lower concentrations of salt, but if they're in a high concentration of salt, they can survive. Right? Then you have your barophiles. Again, phial means love, baro is for pressure. So these are organisms that live at under great pressure. We find these like at the bottom of the ocean, say like Challenger Deep, which is 7 miles below the surface. These organisms are set up to fit high pressure, so that if you were to bring them to the surface where they don't have as much pressure on them, the cells will pop and die, because they will die under normal atmospheric pressures. You need to have them under high pressure for them to survive, because that's how they're made. All right, so some relationships now. We can have symbiotic, which means living together, or non-symbiotic, not living together. 3 main types. The symbiotic, the mutualism, that means these are ones that benefit each other. You can have them obligatory or non-obligatory, but these were both species, not just 2 organisms, 2 different species live together, and it benefits both of them. Commensalism is where they live together. One species benefits, the other species doesn't benefit, but it doesn't get hurt either. And then parasitism, where one species is benefited and the other one is hurt. So plus plus, plus neutral, and plus minus is an easy way to think of it. And those are your symbiotic relationships. Your non-symbiotic relationships, you have synergism and antagonism. Synergism is you have 2 separate species that can live just fine by themselves, but if they're living together, they're both better, but they don't have to live together, unlike mutualism where they have to. Antagonism is 2 species living apart, and if they come together, one of them will produce something that will inhibit the other one or stop the other one. And we'll give examples of all these. So again, the mutualism, both members benefit. Obligatory, you have to have this. Commensalism, one's harmed, one's not. Or excuse me, one's not harmed and one is benefited. Parasites, one harmed and one is not. Here are some examples. They have some mutualistic ones up here. So the one up here on the left, this is some bacteria that lives in the nodules of the roots of plants. They both benefit from this. I'm not a plant person, so I don't know a lot about it, but I do know that they can't live without each other. Over here, this is like coral and jellyfish, 2 different organisms that live together, and if they get separated out, they can survive for a little while, but they do end up dying. That's something that's happening in our oceans as the water gets warmed up. The little dinoflagellates, what they're called, that give them color, live inside coral. It gets too hot and they leave the coral, and it causes the bleaching of coral. And if the dental flagellates don't come back in a certain amount of time, and I don't know what that is, the coral ends up dying. And that is happening in our oceans from global warming. So this is mutualism, where they're both together. They benefit each other, both of them do. Here we have commensalism, where you have some algae, the little green pellets here, and the big thing is protozoa. They're both perfectly fine living by themselves, but if they live together, they can both benefit out of this. All right. And over here, you see the stringy things, the fungi and algae or grass. It's a grass. The grass feeds the fungi, and the fungi protect it from drying out and from insects, stuff like that. So mutualism on there. These appear obligatory. These are not obligatory. And down here, because I said this was commensalism, but this was not commensalism. This is non-obligatory mutualism, and this is obligatory mutualism. And down here we have commensalism, where one species, this right here, this is a bacteria, I can't remember the names of, but one benefits and the other doesn't care if the other one's there or not. Over here you have a mite. Yeah, it's a mite and a bacteria that live together on skin, your eyebrows, I think it is. Again, I don't know a lot about it, but the mite does not hurt the human at all, but the mite has benefited from it. The same with some of these bacteria. Doesn't hurt the human, but it benefits the bacteria. So this is commensalism. Then down here is where you have parasites. This is Sinombre, I think this one is the hantavirus, causes problems in there. And so I'm saying, yeah, it's the parasite, so it's benefited, but the host is harmed. Over here, a mosquito. In this case, they're showing you the protozoa inside of it that causes malaria. It's benefited, and the host, the human in this case, is harmed. So that's parasitism. Now your non-symbiotic ones, so your free-living ones. So they don't need to be in a symbiotic. Again, synergism, they cooperate to produce something, to make it better for both of them, but they don't need it. And then your antagonism, where one is going to hurt the other one. We'll get to both of them here. So the examples. Right here, Cellulomonas degrades cellulose, turns it into glucose. So glucose is used by the Azotobacter bacteria. It benefits it. It can survive without the cellulose. But the Azotobacter then releases, it's a nitrogen fixture, releases ammonium here, which benefits the Cellulomonas. Now again, it can survive without that. Both of these species can survive without each other, but together they are better. All right, but they don't need each other to survive. Let's come down here to the ants. That is the leafcutter ants. They grow fungi because they have to survive on this fungi, but they have a bacteria that live on them that produces antibiotics. All right, so antibiotics is the best example of this here. Is right here is the antibiosis. In antibiotics, we get most of our antibiotics come from bacteria or fungi. And what it is they're going to secrete something that's going to stop the growth of other things. So that's that antagonism. It's two separate species. They get one's going to produce something that's going to inhibit the other one, and that's where we get our antibiotics from. The human body has got all kinds of microbes on it, from fungi, bacteria, and protozoas, and this is our normal flora. And it is, we have commensalisms, we have some parasitisms, we even have some synergistic ones that benefit us, but we could survive without them. Now, biofilms. Most microbes live in some kind of a body. They dominate the structure of most environments. And biofilms are where microbes attach to a surface and they kind of stick together also. It's usually from the glycocalyx, the slime layers and capsules help them stick together. And they release chemicals to attract others and to grow more, and they get to a certain level, a certain population, then they kind of stop that, but they work together, and they get to a certain quorum is what it's called, and so it's the quorum sensing. And in this process, they'll release chemicals that turn on certain genes in them, so that microorganisms are too small to really engulf a lot of stuff, especially a large organism. So they work together to like secrete enzymes that'll break down an organism so that they can consume the broken down stuff. But they work together. They can go do better as a group than they can as an individual, even though each individual microbe in it can survive without any of the others because they are single-cell organisms. But these things dominate the world. You find it again. You find biofilms, the slime layer on a pool, the ring around your bathtub, plaque on your teeth. Those are all biofilms. In the wild, most biofilms are multi-species. In the lab, we kind of generally study single-species biofilms. In the wild, most of them are multi-species biofilms. And again, they're showing you that free-swimming ones lose its ability, its motility, it settles down, sticks to it. They make that matrix, which is usually a slime layer or a capsule or something, to stick to it. Once it gets to a certain density, they release molecules to coordinate responses to, again, like I said, for eating food. That's what they're showing you up here. And what it does is it, again, once they get a certain size, they will turn on genes for specific functions. And so they work in unison to digest food particles. And that's just an example of it. All right, so microbial growth occurs at two levels, because growth of an individual cell to get bigger itself, and then to split and become two different cells. And we call that binary fission. It's similar to mitosis, but there are differences in it. So we don't call it mitosis when they do this. We call it binary fission, or binary fission is what we call it. Again, the parent cell has to enlarge first. It's going to duplicate all its ribosomes and anything else it needs in the cell. It's going to duplicate its chromosome, and then it'll split down the middle and make two new daughter cells. There's the process. So you have your individual cell, it gets bigger. Notice that the number, if you were to count these little blue dots, which are the ribosomes, it's made more of them. Now it's copying the DNA. That's a magenta ring, pink ring, red ring, whatever color you want to call it. And the copied one is blue now, just to show the differences. It'll split, divide up the ribosomes and the chromosome apart, and then it'll form the septum in the middle. So it's called transverse fission, binary fission, same thing. It's split into two, creates a cell wall going around it, and you have two daughter cells now. Should be identical unless there's been some kind of mutation or something going on in it. But that is binary fission. Very similar to mitosis, but there are some slight differences. I'm not worried about the differences right now. All right, so population growth. All right, the time it's required to complete that binary fission is called the generation or doubling time. So again, the time to go from one cell to two cells, the doubling or generation. And that varies from species to species. Some of them can be minutes. I think as little as 20 minutes is some of the fastest ones, to days, again, depending on the species. Since the species, the bacteria, microbe will double its population every time it divides. That's called an exponential growth. So here you start with one, slowly, and then eventually it gets really high, really fast. because you double every time. All right. And that's what they're showing you over here. See, 2 to the first power, second power, third, fourth, fifth, and so forth. Your numbers just get bigger. So that's called exponential growth. All right, here's a simple formula to figure this stuff out. If you know the, first of all, you have to know its generation time. You got to know how many you start with. So your Ni, N is the number of cells. So Ni is the initial number of cells you start with. You times it by the generation time, which is the number 2 times whatever, how many of the generations it goes through. All right, and that will give you your final your total, again, the final number of cells for that amount of generation time. Don't worry about memorizing this. I'm not going to have you memorize it. It's simple math to do this. Here they're showing you, they've taken a diluted sequence down. They're showing it in tubes and growing. When you first start inoculated in there, it doesn't look like there's anything in there. And so this one here, it's after 60 minutes, you've got a little bit. 120 minutes, you're starting to see more. 3 hours, 4 hours, or 4 hours, 5 hours, 6, so forth until it just keeps growing. And you just can't see through it or anything. That's the plate counting of it. So a population growth curve. This is on a closed system now. A closed system is, think of the tube, you screw the cap on tight so no more oxygen gets in there, no more water, no more food, anything gets in there, but nothing leaves either. So you're going to start this. When you first inoculate it, we go through a lag phase. And that's because the bacteria or whatever microbe it has to adjust to that system. So it grows, but not real fast. And once it gets situated and going, then it'll go through an exponential growth phase, which is also known as a log phase, not a lag phase, but a log phase, as in logarithmic. And this is where cells you're getting your maximum growth because there's plenty of nutrients, plenty of water, plenty of oxygen, if it needs any of those things, oxygen or carbon or whatever it is. The environment's perfect for it. So you're having by far more cells being produced than are dying. And that happens for a little while. Then you go to what we call a stationary phase. And at this point, you're starting to run out of nutrients a little bit. So the number of new cells is equal to the number of cells that die. So you end up at it flattens out. All right, and you're seeing this down at the bottom. See the red here at the bottom? Those are the dead cells. The green are the living cells. So it's, you have your maximum growth at this point. It goes clear, got a little bit greener, and now it's really green because you get your maximum number here. Now remember, the nutrients, the stuff that you need to survive, you're starting to run out of this. But just like with all organisms, they give off waste products. Over here in the exponential growth, there's not very many waste products. Now we're starting to build up waste products in here, and that's why you have the same number of new ones as ones that die. So it balances out. Our last one is what we call our death phase. And there are limiting factors are going to occur in this. In other words, we're running out of our nutrients, our resources that we need, but we've accumulated lots and lots of our waste products, our toxins, things that we don't want. So now more cells are dying than are being produced, so the population goes down. And depending upon the species and everything, that'll affect what happens at the end. Either they all die, or they'll produce some spores and survive that way, and wait until the conditions get better again. That varies. So this part here is where more dying than are being produced. It's called the death phase. Remember, this is a closed system. Nothing new is going in, and nothing is coming out of it. All right. So how do we monitor this stuff? We can do turbidity, which is very simple. This one is very clear. You can see through it because there's nothing growing in it. This one is very turbid or cloudy because there's lots and lots of cells in it. So they have turbidity meters that shine light through it. And based on the amount of light passing through, we can tell you approximately how many cells there are to give you the population size. That's, again, that's a turbidity meter. We'll do something like this in micro labs. Another one is where you can actually go and count it. Now, you're not going to go and count each and every single little square. What you do is you take samples. So on this item, on this enumeration plate, you're not going to count everything. You're going to do a sample of it and estimate the population from that point. You can do it manually where you're actually looking at it, or they do have automated systems. That is it for Chapter 7, Environmental Factors. See you later.